Hello students, today we are going to study DNA replication in prokaryotes. DNA replication is the biological process where a cell makes an exact copy of its DNA before cell division. This ensures that every new daughter cell gets a complete and identical set of genetic instructions. DNA replication takes place during the S phase, synthesis phase of interphase in the cell cycle. Deoxyribonucleoside triphosphates serve dual purposes. In addition to acting as substrates, they provide energy for polymerization reaction. The two terminal phosphates in a deoxynucleoside triphosphates are high energy phosphates, same as in case of ATP. Before we understand DNA replication, let's meet the molecular machines that make it possible. First, helicase. Helicase unwinds the DNA double helix by breaking the hydrogen bonds between complementary nitrogenous bases. Second, DNA-dependent DNA polymerase. This is the main enzyme responsible for synthesizing the new DNA strand. It uses a DNA strand as a template and adds complementary deoxyribonucleotidase. Importantly, DNA synthesis always occurs in the 5' to 3' direction. These enzymes are highly efficient enzymes as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 million base pairs, compare it with human whose diploid content is 6.6 million base pairs, completes the process of replication within 18 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymerases have to be fast, but they also have to catalyze the reaction with high degree of accuracy. Any mistake during replication would result into mutations. Finally, DNA ligase joins adjacent DNA fragments by sealing the breaks in the sugar phosphate backbone, especially between Okazaki fragments on the lagging strand. In the process of replication, firstly, the helicase enzyme unwinds the DNA double helix and forms replication fork. For long DNA molecules, since the two strands of DNA cannot be separated in its entire length due to very high energy requirement, the replication occurs within a small opening of the DNA helix referred to as replication fork. The DNA polymerases on their own cannot initiate the process of replication. Also, the replication does not initiate randomly at any place in DNA. There is a definite region in E. coli DNA where the replication originates. Such regions are termed as origin of replication. The DNA-dependent DNA polymerase catalyzes polymerization only in one direction, that is from 5' to 3' end. This creates some additional complications at the replicating fork. Consequently, on one strand the template with polarity 3' to 5'. The replication is continuous, while on the other the template with polarity 5' to 3', it is discontinuous and short stretches of DNA is produced called as Okazaki fragments, which are later joined by DNA ligase. Why do we have a leading strand and a lagging strand during DNA replication? The answer is simple. The two strands of DNA are antiparallel, meaning they run in opposite directions. But DNA-dependent DNA polymerase has one strict rule. It can synthesize DNA only in the 5' to 3' direction. So, on one template strand, which runs 3' to 5', DNA polymerase can continuously synthesize the new strand towards the replication fork. This is called the leading strand, but on the other template which runs 5' to 3', continuous synthesis towards the replication fork is not possible. Therefore, DNA is made in short sections called Okazaki fragments. This is the lagging strand. Later, these fragments are joined together by DNA ligase. So remember, leading strand continuous synthesis, lagging strand discontinuous synthesis. And the reason DNA polymerase can synthesize DNA only in the 5' to 3' direction. Now it's time to test your understanding. Try these important questions. These questions are important for your CBSE and NEET preparation, so practice them carefully. And if you found this video helpful, Like the video, share it with your friends, and subscribe to the channel for more biology concepts, important questions, and exam preparation videos. Keep learning, keep practicing, and keep growing.